Thank you, Mary. Welcome, everyone. We have a more or less a steady list of participants. We can go on. I think this is the session for metabolic and knowledge engineering, part 1. Then we will have the second part later today, as Mary said before. And let's get started without more talk. The first presenter will be Ellie Brown, which is a senior in the biological engineering program at Utah State University. She's working with Dr. Zhang and Jixun Zhang's lab. And she will be presenting the work modeling of the engineering production of curcumin in E. coli. Ellie, when you want, you can start sharing your screen and get going with your presentation.
You'll allow me to share my screen, so it's disabled.
Dimitri?
Okay, let me see if I need to make you a host. Usually, okay.
Yeah, it's disabled. Okay.
Please check now and see if you can do it.
Right. Yeah, perfect. Thank you, Dimitri.
Everyone see my slides?
Yes, perfect.
So like he said, my name is Ellie Brown. We are from Utah State University in Logan, Utah. I worked with Michael Cotner, Dr. Zhang, and Dr. Jixun Zhan, and we worked on the modeling of the production of curcumin in E. coli. So what I'm going to be talking about today is the introduction and motivation of why we wanted to do this, why we wanted to create this model, how we created the model, and then our results and discussion from that and our future work. So first, this picture is turmeric. It's a common spice used in foods like curry. It's also been a medicine traditionally for thousands of years. So the molecule that we're interested in turmeric is called curcumin. It's the major bioactive product in turmeric. It has a variety of biological activities, such as these listed here: antioxidant, anti-cancer, anti-allergic, anti-inflammatory, and anti-Alzheimer's. So our motivation for doing this is that currently curcumin production relies on growing of the turmeric and then extracting it from the plants. This requires a lot of land, manpower, machinery, water, nutrients, organic solvents, time, all of those things that cost money, really, and time. We want to make curcumin in a bioreactor instead, because instead of many weeks for that turmeric to grow, it will take only 48 hours to grow the curcumin. Also, the product is much more pure and constant in the lab in a bioreactor. And our goal is to grow this curcumin in E. coli, which is the microorganism we've chosen. Then modeling the pathway before growing it in a bioreactor saves even more time and material, because we can change the parameters that we want. For example, the concentrations of the enzymes in the model to see what it does to the yield of curcumin, and then we can go use those parameters that we've decided on in a bioreactor and get our desired result out. So our model predicts the production of curcumin in E. coli. This is the full pathway from L-tyrosine to curcumin. So first, L-tyrosine is converted to p-coumaric acid via TAL, tyrosine ammonium lyase. And then p-coumaric acid can either go to caffeic acid or p-coumaryl-CoA with either C3H or 4Cl. Those both can go to caffeoyl-CoA with the enzyme switching. And then both caffeic acid and caffeoyl-CoA can be converted via COMT to either ferulic acid or ferruloyl-CoA And then ferulic acid is then converted to ferroyl-CoA via 4Cl. And then ferroyl-CoA is converted via DCS to ferulyl acetyl-CoA. And to make curcumin, the CURS enzyme takes one ferulyl acetyl-CoA and another ferulyl-CoA from the other substrate to make curcumin. So we made our model on Prism. It's a probabilistic model checking language. We've chosen the CTMC, the continuous time Markov chain, as the background and governing model. It's commonly used when modeling biological systems. It just determines how the random process will happen. So we made a couple of assumptions before making this model. Our first assumption is that the model is greatly unaffected by the cell's central metabolism. So malonyl-CoA is required to make ferulyl acetyl-CoA via DCS. So we just assumed that that's in constant sufficient supply because the cell itself makes that constantly. And this assumption just improves the efficiency of our model by decreasing the amount of reactions, thus decreasing the amount of time needed to simulate our model. Our second assumption is that the byproducts of the last two enzymes, DCS and CURS, are negligible. They make 3 more curcuminoids. They're listed in the last bullet point there below. The reason we can make this assumption is that curcumin is the major product and the other 3 are just trace products in the end. So assuming that won't make a huge difference, and these assumptions are made not at the price of the accuracy of our model. So to further explain our model, first 9 species and 10 reactions were used from that pathway before. Each reaction is essentially a probabilistic transition. So I'll show you a snippet of our model in a later slide, but I'll just explain briefly what is happening first. So the rate of the reaction, each reaction has a rate of reaction, and it's the ratio of each of the rates that is the probability of that reaction occurring. And when a reaction is chosen to occur, it causes an update in the concentration. So 1 more molecule of this substrate, 1 less molecule of substrate, 1 more molecule of product. And the reaction rates are calculated using Michaelis-Menten and Hill equations, which are here. So Michaelis-Menten is a very well-known kinetic equation. The Hill equation was used just for the enzyme DCS. This is because in the paper that we got the information from, when they graphed the data for the DCS reaction, it looked more sigmoidal than it did linear. So linear would mean Michaelis-Menten and sigmoidal would mean the Hill equation. Hill equation just adds that this n slope on these 3 variables. This Michaelis-Menten equation does not have. These are the kinetic parameters that we found in literature. They all have Km and Kcat values. DCS down here for the ferulyl-CoA substrate has 1 more. It's the Hill equation slope, n is 1.8. Here's the snippet of the model. So this is just 2 equations that we've taken from the model. The model is many equations to show each reaction. This FCOA. So first FCOA is ferrolyl-CoA, FACOA is ferrolyl-acetyl-CoA. So this is the reaction using DCS, and this is the reaction using CURS. So this first term right before the arrow is called the guard. It's essentially an if statement, and this arrow is looking for true. So if there exists ferrolyl-CoA, then this can happen, has the possibility of happening. Same thing for this, if there is ferrolyl-CoA and if there is ferrolyl-acetyl-CoA, this could happen. This V1 and V2, these are the rates of the reaction. They're usually in equation form in the actual model, but we've kept them out just for space here. So this would be the Hill equation, and this would be the Michaelis-Menten equation. So each simulation is thousands of steps, and each step, the program looks through all of these reactions, chooses the ones that the guards are true, and then sums up all of the reactions, and then the ratio of each reaction to that sum of reactions is now the probability that that will happen. And then the program chooses one reaction to happen based on its probability. So let's say this first reaction happens, then this FACOA prime, that's just the updated concentration of ferrolyl-acetyl-CoA, and it adds on one molecule because that's the product of this reaction. And then ferrolyl-CoA loses one molecule because it's the substrate of that reaction. So it goes through this thousands of times, and then when we run it, it runs 1,000 stochastic simulations, which are then averaged at the end. So this is our graph of 1,000 averaged simulations. On the bottom here is time on the x-axis. This is up to 48 hours simulated, and on the y-axis, it's the concentration of each substrate or product, each species. So as you can see, most of the products and substrates have kind of leveled out once they get to a certain concentration, especially up here. This is ferrolyl-CoA, but curcumin increases linearly throughout the entire thing. And you can barely see it, but on this y-axis, there's a little blue tint that is p-coumaryl-CoA. You can see it better on this, just zoomed out. This little blue line, p-coumaryl-CoA. So it increases much, much faster than any of the other products. So this observation led us to believe that the enzyme using up p-coumaryl-CoA was the limiting enzyme, and that was C3H. So 4-CO makes p-coumaryl-CoA and C3H uses it. So using that knowledge of the C3H being the limiting, we wanted to change the concentration of C3H to increase the concentration of C3H, so it would use up more p-coumaryl-CoA and hopefully increase the yield of curcumin. So we then did some overexpression experiments. We increased C3H, and then we increased each of the other enzymes from 25 to 50. We just did one enzyme each simulation just to see what happened. And there were some enzymes that increased it a lot, some that decreased it, interestingly, and we marked those for future optimization. This is our data from that overexpressing experiment. So this top row is just all the enzymes at 25 milligrams per liter. This is the yield of curcumin at the end. And then each enzyme was increased to 50, is doubled. And this is a percent change from this initial. So when we increased C3H, it had a 33% increase of curcumin yield, which is quite a lot. And we expected that, so that is great. DCS, however, increasing that, sorry, excuse me, DCS decreased the curcumin yield. And we think that is the case because DCS uses up ferrolyl-CoA and changes it to ferrolyl acetyl-CoA. But then ferrolyl-CoA is also needed to synthesize curcumin. So CURS just had less and less ferrolyl-CoA, so it could not make curcumin as fast. So we will definitely in the future focus on increasing C3H and decreasing DCS. Our future work in the next few months will be continual over and probably under expressing of a few enzymes to optimize our model. And then once we get desirable results, we get a higher yield in the curcumin from our model, we will move that to experimental validation. We'll do it in a bioreactor in the lab to see if our model is accurate. This QR code is our GitHub model, a link to the GitHub. Any questions?
I have a question for Ellie. Ellie, in your assumptions, your two assumptions is that central metabolism is not being affected or affecting your model, and you are not considering the byproducts, right? Yeah. Have you performed, because these are very important assumptions, have you performed any sensitivity analysis in case they are in real life affecting the production of curcumin in your organism?
We have not, but we have used the cell central metabolism before. It takes one of our much larger computers to run. So we will do that in the future when we're writing a real paper, published paper. We will use the central metabolism just for this. It takes 5 minutes compared to 24 hours to run it without the central metabolism. And the byproducts assumption is mainly because we couldn't find data for those. So if we do find data in the future, then we will definitely include them. But right now we're not including them.
Thank you.
Remember that you can ask questions through the Slack channel in the chat, or just asking permission for talk directly. In the meantime, I have also a question, Ellie. In the assumptions also, you were mentioning that malonyl-CoA is not a limiting substrate for the metabolic pathway. But if I'm, correct me if I'm wrong, but I think malonyl-CoA is a substrate for the C3H enzyme reaction from p-coumaroyl-CoA to caffeoyl-CoA. Is that right? And so it would be a substrate for that reaction that you were interested in, the one down here. Then not overexpressing the enzyme as you were proposing may have maybe it will make that assumption to not be true anymore, because you will need more malonyl-CoA to be available for that reaction to happen in a higher rate. So have you planned, or do you have an idea of how to deal with that? Because overexpressing the enzyme may solve the problem that you were mentioning, but you may have the extra added problem of needing more amount of malonyl CoA.
We have not addressed that yet, but that will be in our future work. We'll look into that more. So currently, we haven't looked into that. Sorry.
Great. And there is a question from the chat from Lars. It says the Michaelis Menten kinetics assumes saturation of the involved enzymes. Have you checked if that assumption is fulfilled for the concentrations at the enzymes at which the enzymes are expressed?
I don't believe we have. So we looked through scientific literature to find the kinetic parameters, the Km and Kcat, and then we just started out with all of the enzymes at 25 milligrams per liter. That's all we did at the beginning. So this is not a final model. We will be doing more editing and more optimization in the future. So I don't believe, Lars, that we have assumed saturation of the involved enzymes.
Okay. I think there is another question from Chris. Nice work. You show the simulation results. Are you also doing or planning to do probabilistic model checking?
So this Prism is a probabilistic model checking language. But once we get some more optimization and editing of the model, then we will go validate it in the lab with a bioreactor. I hope that answers your question. If not, let me know.
Great. Is there any more questions? Let me check the chat here. Okay, if there is no more questions, we can thank Ellie again. Thank you for the presentation, and we can move on to the next presenter. Alexis put the link of this in the chat if anyone is interested and didn't have time to take it. The next presentation will be given by Dimitris Papamichail, and it's about Codon-Optimized Degenerate codon set design tool that they have made. Dimitris is an associate professor of computer science at the College of New Jersey, and he specializes in applied algorithm design with applications in computational biology, music arrangements, and other fields. Please, Dimitris, you have the floor.
Okay, hello everyone. I will be talking about a couple of tools that we have created at the College of New Jersey. We call them the codon calculators, and I will explain what that means. It's a little encrypted. The purpose of these tools are to design degenerate codons in order to code for any given amino acid set. And I will go over this process and what it means and why we need it. So once again, yeah, my name is Dimitris Papamichail. I work at the College of New Jersey. This work was primarily done by undergraduate students. I mean, we only have undergraduate students at the College of New Jersey, and my brother helped a little with the algorithmics. All right, so the purpose of this research is to aid researchers and life scientists that are trying to design protein mutant libraries. These libraries usually consist of a specific protein product, some amino acid chain, at locations of which people want to change some of the residues. They want to try different amino acids and see if these versions improve some of the properties of the proteins that they have in hand. Usually these kind of techniques work well with assays that can evaluate a large number of constructs at the same time. High throughput assays usually are the target of this kind of libraries. And there are quite a few groups that work on this and quite a few purposes why they do it. One particular group that we are collaborating with in Princeton, headed by Michael Hecht, tries actually to find out the space of proteins that folds in specific ways and fold efficiently. So I have a couple of examples here in this slide. So given some target amino acid sequence, we select some positions. Well, we decide to vary the residues at these positions. For example, here on the one on the right, we try to put arginine and lysine like in this position here, or these 4 amino acids in the second position, etc. You can see that if we want to create some oligo, and I'm assuming most people here would know about techniques like the Gibson isothermal assembly, where you can take oligos and put them together and assemble them to create the DNA sequence which will express to become the protein. If you want to make changes at 2 locations, you have to create all the combinations of these 2 locations. All right. So, for example, here, if I want to create, let's say, this protein product here, and I want these 2 amino acids to vary in this position and these 3 amino acids at the other position, I will have to create like 6 different oligos in order to do that. All right, in order to be able to talk about these changes that we perform at different locations, we introduced this concept of degenerate bases, which are basically DNA bases which can vary. So we use these letters, like N, for example, we use to signify A or C or G or T, etc. So we have all these codes that can tell us whether some DNA base is changing at some location of the DNA sequence that we are constructing. And when ordering oligos at companies that construct them, it actually costs a little more to specify some bases to be degenerate. So it is relatively small additional cost per base. So if, for example, one base costs like 10 cents, putting a degenerate base may cost 15 or 20 cents per base. So it is reasonable quite often to incorporate some of these degenerate bases in order to get some variation at some locations the DNA sequence that we are ordering. And in reality, that means every time we order some degenerate base, we're getting a bunch of oligos. Like if we have one oligo where one position is an N, that means we're getting actually 4 oligos. Okay, one oligo that has A at that position, one that has C, etc. All right, so using these degenerate bases, we can construct what's called degenerate codons. Here I have actually one example from the group of Michael Hecht at Princeton, where they took some protein and they took some codons here and they put some degenerate codons, which are made again of degenerate bases, which means each of these codons code for a number of different codons and therefore can encode different kind of amino acids. One example here is this codon NDT, which means the first position can have A, C, G, or T. The second position here can have A, G, or T. And the last position is the base T. That means this degenerate codon here codes for these 12 different codons. And these, how many are these? Like 9 or 10 different amino acids. So it is possible to insert this kind of degenerate codons in the DNA sequence you order and get a number of different products, basically protein products out of this DNA sequence once it's expressed. Now, there are many reasons why we will do that. And in this case, they were trying to modify some area of this protein and see where it behaves better when it folds or binds some ligand better. All right, to aid this process of designing degenerate codons to code for the amino acids that we want at certain positions, a tool has been created in the past called Codon Genie. This was created by a group in the UK. And surprisingly, right now, it's not available online. In the last year or something, they took it off online. But I believe it is open source, so you may be able to download, actually install it in your own machine or a web server. So in any case, the purpose of this tool is that someone selects some amino acids that they want to code for, and they want to find a single degenerate codon that can code for all these amino acids. So once you select all the amino acids that you want, the tool will find several different degenerate codons, which we can see here in this column in gray. And for each of these, it will tell you the amino acid that it codes. And for example, in this case, the 7 amino acids that we selected, all of them are coded by all of these degenerate codons. But on top of that, these seem to be coding for some other amino acids that we may not want at this position. Also, all these amino acids, all these degenerate codons here are ranked by some score, and I will explain what the score is. It has to do with codon bias. And of course, it tells us here how many different variants we have, how many codons each of these degenerate codons actually codes for. So Codon Genie ranks these degenerate codons by some codon score, which is defined, and here they have a lot of formulas, but it's a very simple concept actually. It's just the arithmetic average of the individual scores of each codon that they have there. And the individual scores for the codons are basically frequency ratios of these codons that appear in the organism, in all the genes of the organism of interest, over the most frequent synonymous codon. So for example, let's say, let me see, there are some questions in there. Okay, yeah, codon gene, excellent. All right, so just to explain what, for a single codon, what this score is, if we take the frequency, let's say one codon appears, coding for some specific amino acid appears 20% of the time, every time this amino acid appears. And then another codon, let's say the most frequent codon is 40%. That means this codon, the score will be 0.5 because the ratio of like 20 over 40 is 1/2. Okay, so the most frequent codon basically will get a score of 1 here. And the least frequent codon will get a score that is going towards 0. Okay. And if we take the arithmetic average, we get that score that you see here in this column. Do let me know if you have questions, at any time, actually. All right, so the thing that we noticed is that when you're designing degenerate codons using Codon Genie, you are only restricted in using one degenerate codon at any position. And that leads quite often to design degenerate codons that code for many undesirable amino acids, which means the library size that you will create will be much larger than the one that you want, and will contain a large number of the variants in this library will actually be unwanted, will contain amino acids at positions that we don't want to see. So we thought of ways actually of alleviating that fact, of making some improvement. And we created, this was as a concept last year, we created a tool that now given a set of amino acids, it will find not a single, but multiple degenerate codons that code for these amino acids, but exactly for these amino acids, with no undesired amino acids included. Not only that, but this tool will find the minimum number of degenerate codons that can perform that task. So, for example, in the previous example that we saw with Codon Genie, selecting these 7 non-polar amino acids over there, the Codon Genie suggested a degenerate codon, single degenerate codon that codes for another 5 undesirable amino acids. Our tool will tell us that these 7 amino acids can be coded actually by 2 degenerate codons, for example, DTB here and GBA, which code for the exact 7 that you want and nothing else. Okay. All right, so this first version is a proof of concept. And this example that it shows here on the bottom is just created randomly based on the process that we figure out how many degenerate codons are needed for each amino acid set. So it's doesn't have any specific properties. So this may be of somewhat limited use when someone actually wants to use these degenerate codons in their designs. So we decided to follow the same route that the Codon Genie people took, and rate the codons based on some score and based on some organism of preference. So we added to our tools here an organism tab. We currently have only 3 organisms there: human, mouse, and E. coli, but we can increase them arbitrarily as needs change. And by selecting the organism now, when you get the optimal number of degenerate codons to code for a specific set of amino acids, you will also get one example here that is optimal based on this codon score. So this, for example, DTK and GBT are two degenerate codons that have the best possible score coding for these 7 given amino acids. Oops. All right, so we discussed with our collaborators and they saw what our tool can do, and they asked us the following. Now, it happens that this group especially has quite often a large number of mutations around the same area, which means that many of these mutations end up in the same oligo instead of being distributed among the oligos. So if these were distributed among the oligos, it wouldn't be a big problem because the synthesis costs, although they increase, they don't increase dramatically. But once you start having many positions close by that you need to have several amino acids at this position, then you start multiplying the number of degenerate codons you have here in order to figure out how many you have to order in order to synthesize this library. In this example here, in order to have to code for these amino acids at these 3 positions, I will need the first position here, I will need to use 3 degenerate codons, the second I will need to use 4, and the third 4. That means I have to order for only these 3 mutated positions here, 48 different oligos in order to synthesize the library that I want. And of course, these, as you can see, increase exponentially with each additional location that I will add some number of degenerate codons. So although this whole project started in order to reduce costs, because when you have a library that's not specific, quite often you have to spend a lot more time trying to figure out the variants of interest in this library. Therefore, you're spending a lot of money and a lot of time of your postdocs and your graduate students trying to figure out what's going on with this library. And these costs usually were much higher than a slightly increased synthesis cost. But once you want to create this kind of oligos, which increase exponentially in number, now the synthesis cost can become also significant, again, and considerable. So the group asked us to try to compromise between library quality and the cost to synthesize, to order these oligos and put them together. So what we did is we added an option to our tool. When you select some number of amino acids and some organ in here and you click submit, you will get, let's say in this case, the 4 degenerate codons that are the minimum number in order to code for these amino acids without coding for anything else. But also you will get some optimal subsets and supersets. What this means is that since this set of the amino acids needs 4 degenerate codons at a minimum to be coded exactly without any undesirable amino acids, what can we do with 3 degenerate codons? So what is the maximum cardinality subset? Like what is the largest subset of these amino acids that can be coded by 3 degenerate codons, or by 2 degenerate codons, or by 1? So these are the options that we present here. And again, we select degenerate codons here that optimize this codon measure that we talked about before, this codon score. And the same thing we do with supersets. So we want to find the smallest superset that is coded, sorry, the smallest superset of these given amino acids that's coded by 3 degenerate codons, or 2 degenerate codons, or 1. Now, some places here I have this null value because I'm only exploring supersets and subsets that differ at most at 3 residues from the original set. So if we start having differences of 4 or more amino acids, I'm not considering these options at all because we're going too far away from the desired amino acid set. And yeah, the links, I think they have been placed in the chat. So actually, let me go and show you the tool. So the tool looks something like this. Actually, I don't know why, but the student of mine that coded this at the beginning liked plain flashcards, I think, with letters here. So if you see W, you have no idea. I have no idea what amino acid it is until I press it, and then it tells me it's tryptophan. So you can use this also to learn the single letter codes for amino acids. Right? It's a very useful tool. All right. So once you select some amino acids here, you select an organism of preference. You click submit. So you get the information that this, for example, set of amino acids will need at least 4 degenerate codons to be coded exactly. But we can actually, with 3 degenerate codons or 2 degenerate codons, we can code for subsets and supersets that differ by at most 3 residues. And you can see here which degenerate codons can do that task. I think the tool is relatively easy to use, but let me know, I mean, if you have questions on how this is done. Oh, we find one with 5. Oh, by the way, yeah, we also found that 6 degenerate codons are always sufficient to code for any given amino acid set. Now, the algorithmics and how we did all things, these things, yeah, I'm in the process of writing a manuscript, so it will be published in case you are wondering, because it was a relatively hard task to figure out and prove that this is the minimum number of degenerate codons that can do it. But it didn't fit in the pages of this abstract. All right. Let me see. And of course, the classic disclaimer on all my software that I have only proved that this is correct, but I have not tried it. So, let me know if you find any errors in the code. I will appreciate it. Okay. I would like, of course, to thank all the students that got involved in this work. The HPC administrator, which very nicely has given us a virtual machine to host this web server there, and an NSF grant that actually funds this cluster in our work. All right, thank you. And do you have any questions?
Thank you, Dimitri. I'm checking if we have any questions. In the meantime, I have a question. At some point, you mentioned that there is a trade-off between the, let me remember, yes, the quality and the cost of the synthesis. Have you, or maybe it's the way you solved it, but I don't know the details. Have you tried to state this as a multi-objective optimization problem with the integrals and stuff like that? Or maybe the question should be, how are you solving this trade-off between quality and cost?
Well, now it's a human-generated solution, basically. I could, and it will be a relatively easy task. Yeah, I'm not getting involved with integrals or this kind of optimization, but I optimize algorithms. So for me, it will be relatively simple to, for example, maximize the number of interesting constructs versus the cost of the library here, because I just have to find positions here where I can maximize the number of amino acids that are encoded by minimizing the number of degenerate codons that I'm using. And that's a very, very simple function so I can do it discretely, algorithmically. I don't need to involve any like analysis optimization methods.
Okay.
But anyway, right now, the collaborators are doing this by themselves. So they judge, like in some positions, they get too many degenerate oligos. And they can decide, okay, this position, let's say what I can do with 2 degenerate oligos. How many of these amino acids can I code, for example? Or how many extra will I get? And they do it by seeing and adjusting. And our tool makes it relatively fast for them to be able to figure it out.
Okay, in that case, then having the list that you have at the end of what you get, using less degenerate codons, you get what you can do with that.
Right. By reducing the number of degenerate codons here, I'm reducing the number of oligos that we're ordering, the total number of oligos being ordered. And then by providing both subsets and supersets, the researchers can decide whether they want to reduce the size of the library of useful variants in the library, or they want to increase the number of non-useful ones. Right, and undesirable ones. So they can play with these 2 parameters to find what they're comfortable with, basically, the numbers that they believe that their assay later can deal with.
Okay, thank you. There is a question now from Lars in the chat. Very nice. The question is, have you incorporated some sort of limit for the codon quality? I'm thinking of a scenario when it would be better to use 3 degenerate codons instead of 2 to get a better quality of codons.
That was exactly the question I was answering now. So right now, we're not doing that automatically. although again, it can be relatively straightforward to do it automatically by saying, if you specify, for example, what is the largest amount of the undesirable constructs that you want, and what is the minimum size of useful constructs that you want, I can tell you how many oligos you will need, for example. That is relatively straightforward for me to implement that. Okay. By the way, this is part of a bigger project where actually, yeah, one person gives you the whole protein and the locations they want to vary. And we have an algorithm that actually will decide how to size the oligos and where to make the overlaps in order to minimize, again, the number of oligos that have to be ordered. So we will place like these degenerate positions, for example, avoid overlaps, etc. So at the end, we will have a tool that, yeah, you can specify all of these constraints that you have and will produce directly for you the order for the company. It will tell you which exactly oligos you want and how they will be assembled once you get them back, such that you get the desired library that you want.
Great. Awesome. I don't know if we have more questions. Let's check in the Slack.
Yeah, please ask more. You are giving me ideas for more cool algorithms in the future.
I don't have any more.
All right. We'll have more extra time for coffee.
Anyway, if someone has more questions for either Dimitri or Ellie, it can be done in the social or later with other means, with Slack and email. Okay. Thank you very much to the presenters, to Mary for the introduction, and to the people that was listening and paying attention to all this.